Started here, I'd like to introduce our keynote speaker today. Paul Sisco has spoken to us before. He was the first science coordinator down in the Carolinas more than 10 years ago. He was a geneticist at Meadowview Farm, and his background is from Cornell. And he's come up and spoken to us and has always been a straight shooter and calls it how he sees it, and that's always been valuable to us. We've been doing this work for a long time. So please warmly welcome Paul Sisco.
Need someone to run that, Paul?
Yeah, I think we're...
I'm happy.
Got a button?
So anyway, everybody's been citing their pedigree, so this is mine. Almost 40 years ago, I guess 39 exactly. That's me as a graduate student at Cornell. I took the train from Ithaca, well actually Syracuse, to Urbana, and met Charlie Burnham at the train station. That's the first time I'd ever met him, and we got to be friends, and he signed me up for the Chestnut Foundation in '86 at another meeting. This was May's genetics meeting. I was a corn geneticist, and then was a professor at NC State in corn genetics, and when a position came open at Meadowview, it just seemed like a really interesting thing, so I took it and was there for 3 years, and then moved on to become regional coordinator for various reasons, and I've been retired for 11 years now. So I'm not speaking in any official capacity at all, but I am in charge of the Carolinas seed orchard. I've been working with that for 18 years, so about the same period you've been working in Massachusetts. And I've been making observations, and I have some thoughts that may help you, I think, some practical suggestions, which is what I'm going to try to do today. I really applaud this chapter. When I was hearing all the things that you've done and all these orchards that you've got and the ideas that you've got, it's really a talented and a hardworking chapter, and I really applaud everything you're doing. I'd also like to acknowledge Kendra Collins. Kendra, stand up. Maybe, not everybody will know you. Stand up, Kendra. Okay, Kendra is the regional coordinator for New England, and based in Burlington, Vermont, which is a great town, and has come all the way here. And one of the things I really want to emphasize is that we have an outstanding science staff in the American Chestnut Foundation right now, and they deserve all the support you can give them. I'm really happy with what's going on, and that is definitely not something I'm criticizing. They're doing a great job. But things have changed. This is my third talk to the chapter. I saw that I talked in 2003, which would have been your fourth meeting. And I talked 5 years ago, almost exactly to the day. It was December 6, 1914, and it predicted that we'd be in the situation we are now. So I've been thinking about it. I organized our Carolinas chapter with what I called an insurance policy in case the Burnham Plan didn't work. And describe what that is and what my experience is so far, okay? So this is my outline for this talk. First of all, I'm going to try to describe the Burnham Plan as I understand it, as it was implemented, and why didn't it work, to try to explain some of the reasons why it didn't. Number 3, very important to you folks, because you're like me, you're very, you spend a lot of time and effort making seed orchards. So what do we do with the seed orchards at this point? Number 4, talk a little bit about genetic engineering chestnut trees, which is another tool we have in our pocket now. Number 5, again, to think of some of the issues of philosophy and practicality as we're coming into this. And finally, that's again my exhortation, please support your excellent science staff. They deserve all the support you can give them. This is, I just pulled this out when I was getting ready for this talk. I pulled out the talk I'd given 5 years ago, and I started laughing. I said, hey, I could just give that talk again and I could be really lazy, but things have changed some, but I sort of anticipated what was coming, and I chuckled to myself. I'm not that far off. The way I focused it 5 years ago was in thinking about scientific paradigms. Paradigms are big ways of thinking that scientists have, and sometimes it constrains their way of thinking. And when there's a shift in those paradigms, it causes a lot of disruption in the scientific community. People often think of scientists as you know, as just purely interested in the truth and not affected by emotion, and they just look at their data and they make conclusions, and that's not true. They're emotional like everybody else. And so when they have certain mindsets, it's very difficult to change it. The classic example is whether the Earth goes around the sun or whether the sun goes around the Earth. You know, that was a big change in the Renaissance, and there was a lot of resistance to that. People were threatened with death for saying that the Earth went around the sun. And with this shift in paradigm from the Burnham Plan to a plan which will say the genetics are much more complicated than that, it will be hard for some people to change. And we're going to have some people in the organization that will stick to the old paradigm till they're dead. They're stuck on it, and they're not going to change, and you just might as well know that. One thing I read when I was talking about paradigms is that the only way to get rid of an old paradigm is to get rid of the old scientist. This was the way I described this again 5 years ago in the paradigm shift, that the old paradigm under the Burnham Plan, that resistance to chestnut blight is controlled by only 2 or 3 genes, so that back crossing is an appropriate method, and that turned out to be wrong. The other paradigm that I said at that time, and I was wrong about this, was that, again, our paradigm was that all the isolates of the blight were essentially the same, and that some of them just grew faster than the others, so that there was no specific gene for a specific strain of the blight. At the time I talked 5 years ago, we had some data that indicated there might be some specific genes for specific strains. Since then, Jared Westbrook, our current scientist, tells me that all the data he has supports the old paradigm, that it's just a matter of how fast they grow. There is no gene for gene interaction. And that some way makes sense, because chestnut blight is what's called a saprophyte. It likes dead tissue. It's not specific on living chestnut. So it doesn't have that kind of close genetic relationship to chestnut that some other diseases like rust diseases have with wheat, where there's a very clear gene for gene correspondence. You get one race of wheat rust, you get one gene for resistance, that race overcomes it, you get another gene for resistance, that race overcomes it, and wheat breeding is a constant contest between genetics and the fungus, gradually overcoming each gene as it's brought into the mix with the gene for gene system. Chestnut blight seems to be different, that it's a disease where even on dead tissue, chestnut blight will grow quite well. On Chinese chestnut cordwood, chestnut blight is very happy, will grow all over it. It grows on oak trees. So it doesn't have that close relationship to chestnut that a rust disease has with wheat, and therefore probably the genetics are different. Okay, so what was the Burnham Plan as implemented? Come on, folks. This is the way I diagram it. The way, a little bit previous, it showed you the tree that was gradually filling up with American chestnut. I like to think of the way that the resistance works. And so the way I did it in this diagram was to use a red petunia flower for high resistance and a white petunia flower for no resistance. Okay, this is a petunia. It has nothing to do with chestnut, but it's just a nice color feature to indicate that that's high resistance, white is low resistance. When you cross those two, you get something that's intermediate, which is pink. And a lot of people seem to understand that when you cross red and white flowers, you often get pink. And that's the way blight resistance works. You cross something that's highly resistant with something that has no resistance, you get something that's intermediate. And with the back crossing plan, the idea was that you took a pink, you crossed it back to an American, and so this is where, and I just have here the percentages, you're gradually building up the percentage American chestnut, each generation selecting for something that's intermediate, because that's the best you're going to get. And then in that final two steps, you stop breeding back to American. Instead, you intercross two members of the backcross 3 generation, two pinks. And when you intercross two pinks, you hope to get a red. So that's what the final step is, that you're looking for something that's highly resistant, that still has 94% American or so, thereabouts, and that those reds are the only thing you leave in the seed orchard, so that when they open pollinate, everything that comes out of that seed orchard is highly resistant. That was the Burnham Plan. Any questions about that? Because that's the basis that we've all been working on.
Question?
Yeah.
Is 94% a magic number, or is that just because that's the way we chose that that generation will start doing the process?
It's just that each generation, on average, you should lose half the Chinese genes, and so 15/16 is equivalent to 94%. So now, in reality, because you're selecting for certain Chinese genes, you're selecting for resistance, it's probably going to be less than 94% American, because this is just assuming there's no selection. But since you're actually selecting for Chinese genes, you're probably not going to get up to 94% in the back cross unless you're not selecting very well.
Why is it at BC3 we chose to do F2?
Because it was felt that by that time they were American enough that it would, in essence, be American. Now, there actually, 20 years ago, in our first science review, there's a big argument about that, because our main scientists wanted to go to the backcross 6 generation, which would make 96 or 97. But the question was, again, are you going to be losing resistance through these backcrosses? That was the worry, that the Burnham idea of 2 or 3 genes wasn't correct, and that we were gradually losing resistance, and if we kept going, we were going to lose too much resistance. So that was actually a debate that happened 20 years ago, how far we should go. And again, it's a matter of diminishing returns. 50, 75, 87, 94, the next step is 96, then 97. I mean, you're not gaining much for each generation. Okay, so this was the goal of the Burnham Plan, is implement. To have a Chinese level of blight resistance, to have approximately 95% American genotype, to have enough genetic diversity to restore the species. And this was important, that we were not just trying to create a clone like apple. In fact, I got some of these apples back there are wonderful. McCown's my favorite variety. But apples, you get a clone that you like and you graft propagate it. So if we were just doing that kind of breeding and we got one chestnut that was great and we graft propagated it and put it everywhere, we might have the type that we want, but we'd have no genetic diversity and we would not be restoring a species. So to restore the species, we need to get the diversity and we need to get local adaptation, and that was the whole rationale for the chapter programs. That's why you're going through all this effort that you're going through, is to get those 2 steps in, the genetic diversity and the local adaptation. And in the South, we have an additional problem, which you can thank your lucky stars you don't have at this point, which is to get an additional resistance to Phytophthora cinnamomum. And I have to disagree with Brad, there's no American that has resistance. We have to bring it in from Chinese, and right now we've got 5 genes located for resistance. Okay, so this was the key assumptions. Then I'm going to tell you why they turned out to be wrong. Number 1, that the genetics of resistance to blight was simple, 2 or 3 genes. And number 2, this was added by the pathologist, that the punch inoculation method, which we're using and you guys are using, could reliably distinguish between resistant and susceptible trees. And there's some problems with that method. Okay, assumption number 1, the genetic resistance controlled by 2 or 3 genes. This was the diagram and the rationale behind that. In 1992, when Fred Hubbard first started MetaView, he made a cross between 2 F1s in Connecticut, created an F2 mapping population, and then had that mapped genetically to find out where the resistance genes were. And what turned out in that F2 population were 3 genes popped up, one on linkage group. The 12 chromosomes of Chestnut are labeled with letters, traditionally. So on this chromosome, which is either chromosome G or chromosome 7, whichever you want to call it, there was one there was one on the 2nd chromosome, and one on the 6th, B, F, and G. That was published in 1997 in Phytopathology. Then there was also a backcross 1 population that was never published, but I worked on while I was at MetaView. And there was an additional locus there on E, and we were always arguing whether the one on E and B were the same. There was a lot of arguing going on because we didn't want to, Fred didn't want to add a 4th locus. But anyway, it looked like there might be a 4th one. But that was the thinking that supported the Burnham hypothesis, and so for a while we were quite happy with what we were doing.
Paul?
Yeah.
Just when you say locus, that to me sounds like multiple genes, not a.
Well, yeah, it's a genetic term. Locus is Latin for place, and it just means that there's a place on the chromosome that has resistance. How many genes are in that place? Nobody knows. So that's why geneticists, when they don't know specifically what's at that position, they'll say that's a position that's important. But there might be 6 genes that are tightly linked at that position. Who knows what's going on? It's, I could tell, it's like saying that there's something between Des Moines and Peoria, you know. But where it is in there and how many towns there are, you don't know.
Thank you.
And the goal of the original model then was homozygosity for these 2 or 3 genes. And homozygosity means that you have doses from both parents, so that you have 2 doses of the Chinese on B, 2 doses of the Chinese on F, and 2 doses of the Chinese on G. And that was the reason for intercrossing, to get that double dose. And then again, that assumes, which was the evidence, that these genes are not dominant, they're additive. So 1 copy gives you 1/6 of the resistance, 2 copies gives you 2/6, 3 copies gives you 3/6. That's what we mean by additive. So if you want to get full resistance, you've got to get 6, what are called in genetics, alleles. 6 different versions of that gene on those 3 loci. 1, 2, 3, 4, 5, 6. That's why we intercross. And this was the genetics, again, in terms of how we were selecting. In the backcross generation, 1 of 8 should have 1 copy of all 3 genes. It's just 1/2 times 1/2 times 1/2. In the F2 generation, it should be 1 out of 64 because it's 1/4 times 1/4 times 1/4. And that's why we have 150 trees in the plot, because the idea was if you have 150, you're 1 out of 64 is an average, so that you're likely to capture 1 in 150 trees. And especially if you have 9 blocks, somewhere in those 9 blocks you should capture it. In fact, you may capture it twice in one block and not at all in another block. And there was a question about whether we might actually be transplanting trees between blocks to get them all to have 1 example in each block. So this was the rationale that underlay the Burnham Plan. Any questions? All right, the first problem showed up in 2002, 17 years ago. I was working we were having trouble with our genetic map, and I was working with Europe. I got a little money from the Park Foundation in Ithaca, New York, to cooperate with a lab in Italy, because they had just published a European genetic map with 12 chromosomes. And I was really curious about how their European map compared to our map, which of their chromosomes corresponded to which of our chromosomes. And they were very happy to cooperate with me. They sent me genetic markers for each of their 12 chromosomes. We put those markers on our genetic map, and then we could tell, well, our chromosome B corresponds to their chromosome 11, and so forth. I remember that one. In doing that, we used a new kind of marker called a simple sequence repeat, a short sequence repeat, SSR, which is very good about distinguishing parents. There's a lot of different variants of this marker. In fact, for one position, there were 31 variants, so that you can find out a lot about parentage, because you have an opportunity for really distinguishing among different trees in the field if there's 31 different versions of that gene running around. We put those markers back on our original mapping population, and lo and behold, we found out that 17 of the 102 F2 trees in that mapping family that was published were actually contaminants. They were not part of the mapping population. We clearly showed that the female was right. We had gathered the seed correctly, but there was some pollen contamination in that group that they didn't know of fact. In fact, when we went back and looked at the original data, there was indications in the original data that there was contamination, but the molecular biologist thought that the breeder knew what he was doing. The breeder thought he knew what the molecular biologists knew what they were doing, so they didn't question each other as they should have. But anyway, it was really compelling, the contaminants. And if you look, I've highlighted the contaminants in orange here. What you're looking at here is the most resistant tree is in the lower left-hand corner. The most susceptible tree is in the upper right-hand corner. This is the distribution. So almost all the highly resistant trees were contaminants, okay? Including some that they'd gone to great lengths to.
What's the y-axis? Y-axis?
It's actually just, it's the SG and the EP canker sizes. So all he was trying to do was to spread out the distribution. But the only key feature here is to look from lower left to upper right, from most resistant to most susceptible.
Sounds like your X and Y are the same then.
Yeah, pretty much. Yeah, it was just a way of spreading the data a little bit. There were also 3 contaminants up here in the most susceptible. I assume that those were outcrosses to American, that these were all outcrosses to Chinese. But that was pretty upsetting. The most resistant down here, controls, were actually Nanking, and this was a mahogany distribution. So one of the conclusions was, well, mahogany doesn't have as much resistance as Nanking does, so the best we can get out of mahogany is right here. Whereas if we'd gone to Nanking, we'd go down there. But that was a disturbing finding. And then, just recently, I almost chuckled on this. Again, there's true believers on this 2 or 3 gene hypothesis. So the true believers went back and said, well, we're down to 85 members of that mapping population. They had created more. They created several hundred more members of that mapping population to get a better map. And so they said, well, we can really narrow down those 2 or 3 genes by looking at a much bigger population. And what happened was they didn't narrow down 2 or 3 genes, they added more genes. In fact, there were 4 or 5 more genes when they added numbers to that real members of the mapping population. And so that analysis got talked about and then got dropped because it didn't provide the answer that the person doing it wanted. So this is the revised model to think about for blight resistance. And again, this is just a model. This is not necessarily what the facts are. But that there may be alleles on every one of the 12 chromosomes, and that when we do our intercrossing, some of these we may get homozygous. For example, on this diagram, I put homozygous there and homozygous one other place. I can't, maybe it's, no, it's not there.
E.
See? Yeah.
E.
E. Oh, yeah, E, okay. Anyway, so I just want to kind of give an idea of where we may be headed. One false bit of information needs to be corrected, and I heard it, was that there's 9 genes now. What Jared Westbrook said, which got misinterpreted, Jared said that he had looked at a clapper mapping family. He's looked at all several hundred clappers in our MetaView orchard. And he looked for loci correlated with resistance in clapper. Now remember that both clapper and graves started with a backcross 1 gene, but with a tree. So it was already 75% Chinese and 25% American, which means you had lost 25% of the Chinese genes before you even started. And among those 25% of Chinese genes you lost were probably some resistance genes. So probably neither Clapper nor Graves started out with all possible resistance genes. They'd already been lost in that first backcross. And in specific, which I think Brian pointed out, in that Clapper mapping family, and looking at Clapper itself, he found out that the entire chromosome 2B in this is American in Clapper. And we know that that's, if you remember back at the mapping population, that was one of the most important loci that came from mahogany. So again, what Brian said, it's going to be a very smart thing to start combining your Graves and Clapper and Nanking when you get it into one mega population to get all those genes together, because none of them have all their genes. Yeah.
So just explain the correlation between Graves and Clapper and mahogany.
Okay, Graves was a backcross 1 from mahogany, and Clapper came from a Chinese tree in the USDA breeding station in Maryland, and that Chinese tree was dug up when they abandoned the USDA program. So that's why we cannot go back to the Chinese grandparent of Clapper. We can go back to the Chinese grandparent of Graves because we know it's mahogany. So we can actually tell, he's actually analyzing Graves now, he'll be able to tell, looking at the original mahogany mapping family versus what's left over in Graves, what has been lost in that very first backcross. So again, to go over what Jared said, that there were 9 chromosomes in Clapper that were associated, but some of those chromosomes could have more than one gene. So that's where the confusion has gotten. People are starting to say 9 genes. No, sir. It was in Clapper, only in Clapper, 9 chromosomes, and that's probably not everything. So at this point, we don't know how many genes there are. Okay, that's still to be determined. And just to show you, I've been working the last few years with Phytophthora in the South, and Phytophthora is a lot easier to work with. The genetics are easier. It really kills trees very quickly in a small space, so you can get a whole lot of numbers very quickly. It doesn't have too much environmental influence. It's a really nice disease to work with. And with that, we've been able to figure out pretty solidly what the genetics are. In fact, we've been able to cooperate with Europe, where they also have a problem with Phytophthora and European chestnut, and they've done crosses of Japanese times European. We've done crosses of Chinese times American, and we've mapped the Phytophthora resistance gene in both those crosses, which is 2 different species in 2 different continents, and come up with the same answer. So that gives me confidence that we know what we're doing. And what happens is we've got one chromosome, linkage group E, that has 3 different genes on that chromosome. One's located here, one's located toward the middle, and one's located at the bottom. And then on K, which is another chromosome, there appear to be 2 different loci, again, areas, which may have themselves more than one gene. And the length of these bars just show you the confidence interval that we have, depending on the mapping populations. These are different mapping populations, some from Nanking, some from mahogany. Again, I tried to do a really good genetic experiment here. We did 2,000 plants from 5 different mapping populations, and came up with this answer. So that's what gives me pretty good confidence that we know what's going on with Phytophthora resistance. And Jared has done mapping in Graves, and the Graves that he has in Metaview, and has picked up these 2 loci pretty clearly. In Europe, these 2 seem to be more important. So it may be the Japanese and European, it's these 2 that are more important. Somehow in the United States with Chinese and American, it's these 2 that are more important. Yeah.
So does the actual Chinese pretty much just have those 2 loci? So the Graves get all of the PRR resistance that Chinese have?
The Graves here are the blue and the green. The green is actually mahogany BC1. The blue is a Graves BC3 family based on a BC2 tree. And what we got was the same 3 loci in the Graves and in the original mahogany on E, and then in the on K it was only mahogany itself. So the Graves may have lost this locus on K. For Nanking we got this, and that, but this was only a Nanking BC1. We haven't done any advanced Nanking yet. And again, that was, we didn't start with the backcross 1 with Nanking. Okay, assumption number 2. We talked assumption number 1 was the 2 or 3 gene. We've shut that down. Now assumption number 2. The punch inoculation method can reliably distinguish between resistant and susceptible. And this is the diagram that all of us are working with. We look at canker size of the SG, which is the milder form, EP155, the more virulent form. And the problem is that there's a large environmental effect on blight resistance, making single tree selection problematic. And I get most of my information from Kendra and Sarah, who look at a whole lot of inoculations, look at the same family planted at 2 or 3 different locations, and can tell you that they get different answers at different locations depending on the environment. So we've got a problem with blight that there is a big environmental component on blight resistance. A tree in a good spot is going to be more resistant. A tree in a bad spot is going to be less resistant. And this even happens in China, where in a drought year, a lot of their Chinese chestnut trees come down with blight under stress. And they're actually still breeding for blight resistance in China. We just had a guy come back, John Carlson at Penn State. He said they're not worried about Phytophthora. They never have problems with Phytophthora, but they have problems with blight. And they're actually trying to breed Chinese genes that are more resistant to blight. If we could clonally propagate chestnut, we could get over this. This is done in hazelnut and cottonwood trees where you can, in one way or another, clonally propagate that initial selection in different environments, hit it with your inoculum again, and see if that resistance holds up over different environments. And that gives you more confidence in your original selection. And of course they do this in hazelnut because they're only looking for one tree, right? They're looking for a particular cultivar that they may call Lewis. So they test Lewis over a bunch of different environments to make sure that the resistance to eastern filbert blight holds up. And the way that they do it, this is, I don't know if that shows up real well, but this is out in Oregon State. My buddy Sean Millenbacher works out there. They put a ring of metal around the tree, put dirt, and the tree tends to sprout a lot, and they can take those sprouts and transplant them and then clonally propagate their selection that way and make sure that it's got resistance. And they do that with any selection before they send it out to growers to say this is a blight-resistant tree. But it's troublesome in chestnut. It's very difficult to clonally propagate it. So then we come down to here the revelation, or whatever it's called, I don't know, in April. Jared said that many of the blight-resistant trees in Meadowview Seed Orchard are actually outcrosses to Chinese. That was unfortunate. And so, plus his analysis showed that the best we can hope for through additional selection within a particular group, like Clapper and Graves, is less than 50% of the Chinese level of resistance. That was his calculations at the time. So, this is what I came here for. So what do we do now? Okay, there's several different strategies you can think about to what to do. 17 years ago, when we came up with those original data, I showed you where the 17 trees were contaminants. Bob Leffel, who's a plant breeder in Pennsylvania, said, okay, we've really got a quantitative system, and I'm going to start from scratch. So this is what Bob did. Starting in 2005, he made a lot of different F1s from different Chinese and Japanese parents. He intercrossed to make an F2, selected the resistance in the F2, intercrossed those to make the F3. This is called recurrent selection, where each generation you're selecting for resistance. You intercross the best ones, select again, intercross the best ones, select again, hoping to gradually put together all those different genes into one package, one population, which has high resistance. And Again, they started in 2005. It's been going on, and the TACF scientific staff visited their orchard at the Gettysburg meeting in the fall. I wanted to go, but I had another obligation. I couldn't. And so they could tell you what the current situation is. From what I understand, the crosses by Japanese actually look better than some of the others. But the key here is that Bob is not worried about percent Chinese. He doesn't care if he comes up with something that's 50% or 60% Chinese. What he was looking for was something that was timber type that he could give to the growers of Pennsylvania to put in their little tree plots for timber. So, and again, we'll get to philosophy in a minute. Bob's a plant breeder just like I am. He was trained at Iowa State. Strategy number 2, and from what I understand, and this is Paul's opinion, this is where Fred Hebert and Kim Steiner are coming from right now, is to start from where we are. That we do recurrent selection by intercrossing our best B3F2 and selecting in the F3 and F4. To assume that we've got in all our populations all the alleles that we need, and all we need to do is to concentrate those in a population. And again, I think your strategy of putting your Nanking and Graves and Clapper together in Massachusetts is a great way to do this. Okay? I would really encourage that. The questions, of course, are, do we have enough blight resistance genes left to get an adequate level of blight resistance? But the more different sources of resistance you put together, I think the more likely the answer is going to be yes. Okay? You put Nanking, Graves, and Clapper together, I think you've got a good probability of getting something that's going to be highly resistant. It may take you a couple of generations, but you'll get there. Will we be able to get enough genetic diversity and local adaptation? That's a question, because when you start concentrating this stuff, you're in essence inbreeding. And are you going to be able to get enough diversity, or are you in essence inbreeding that population so it's not going to have a whole lot of diversity? That's one question that could be answered mathematically, but that's common sense tells you that's going to be a problem. What percent Chinese chestnut will we end up with? Again, the more Chinese genes you're bringing in, along with a piece of Chinese next to them, the greater percentage Chinese your population is going to be. So we don't know at this point what we would end up with in terms of percentage Chinese. I think Jared Westbrook's continuing analysis of blight resistance in different mapping populations will provide answers to this. The more we know about what's actually going on in our different populations, the better we'll be able to devise a strategy. And he's only been able to analyze Clapper so far. He's got Graves in the works, and we've got Nanking coming too. Okay, so again, back to this diagram. This is again a theoretical diagram where if you count, I had 15 different alleles. Will that be enough for long-term resistance? That's the question. Okay, strategy number 3, and this is one I'm using in the Carolinas, and I suggest it as something you can use without messing up your program and without causing you too much more work, okay? So this is the one I'm trying to sell to you guys today. And that's the cross back to Chinese method. Cross our best B3F2s back to pure Chinese F1 or BC generations from other sources of resistance to add genes for blight and phytophthora resistance that were never present or lost in the backcross trees. And again, the question here is, will TACF members in the public be comfortable with trees that have more than 5% Chinese? The trouble is, in the Carolinas, we only have Clapper, which does not have any resistance to phytophthora. So I'll have to get phytophthora resistance in from someplace. And one place is to go back to Chinese. Another is if you come up with a really nice Graves tree here in Massachusetts that's got phytophthora resistance, I'd love to have some seed from that, and I'll put it in my seed orchard, which would be another way for me to add it. But when I started 12 years ago, I didn't have that. So, 2012, 7 years ago. So this was a strategy I thought. And in particular, what I did was to put in male sterile F1s into my seed orchard. Now you saw the diagram that Brian presented where you've got your plots of 150 trees, and you had space between them. What I did was to fill in that space with male sterile F1s. And this did a couple things for me. First of all, since they're male sterile, I can leave them in there a long time. They're not going to contaminate the pollen cloud. So I can use them as controls to see how well my other trees stand up to them. Will my other B3F2 selections be as good as F1s? Secondly, I can use these as seed parents from pollen coming in from the B3F2s to add resistance, and especially to add phytophthora resistance, which I'm interested in, to the population. And if I collect seed only from these guys, I can be sure that the pollen came from these, so I can do it by open pollination, not screw up my B3F3s, but still do something additional. And this is what I'm trying to sell to you guys today. I think it would be smart for you to put in some male sterile F1s and anywhere around, it doesn't have to be in the middle of your seed orchards. And the way to get male sterility is to use American as female by Chinese or Japanese as male. And what I'm doing is using different Chinese and different Japanese so that I can get a mix of different resistances in that seed orchard. And I call it my insurance policy. Okay, I'm putting in some diversity and some more genes that may come in handy down the road. I don't want to have to start 15 years from now from scratch. So I'm trying to think, what's the insurance policy? What's plan B? And that's been my plan B. Again, a way to diagram this to make you think about it, I have these male sterile F1 trees in the middle. The pollen is only coming from these B3F2s. So if I collect seed from the B3F2s, that should only be B3F3 seed, because that's the only thing in there. Yeah, I'll do 1 second, I'll answer. But the male sterile F1s, I can collect seed from them. Their parents should have been these guys. Yes?
What Jared has kind of come up with as a similar suggestion
So who has?
is Jared.
Oh, Jared, okay.
Is what he's calling spiking the punch. But basically what we're working on this winter and probably the next few years is going out, finding F1s, BC1s from other programs or that are just around, phenotyping and genotyping those to select additional sources of resistance, and then crossing those likely with maybe some of the lines that don't have those selections, or even some of the good BC3s or BC4s, and putting 2 or 3 plots of these into the seed orchards just to sort of mix things up a little bit and add some more resistance. What's your opinion comparing and contrasting? I mean, it's very similar to what you're doing.
Yeah, I
it's
think
not
it's exactly the
focusing
same.
on
In fact,
sterols, but
let me go back here, Kendra. What Kendra is saying is that Jared has sort of got along with the same idea, calling it spicing the punch. Is that it? Spiking the punch. Yeah, my grandmother used to have eggnog, and I watched that stuff get spiked all the time. Every person that would pass the eggnog bowl would add a little bit more whiskey to it. In fact, he has the opportunity to do this at Metaview, which I've pointed out to him, because these pseudo F1s that showed up at Metaview should be male sterile.
I think it'd be an interesting population to work with.
Pardon?
I'd like to see us work with those a little bit.
Yeah, and again, my idea here initially, Kendra, is to put those out in demonstration orchards where, like at Biltmore, where we're trying to show something good, and showing dead trees doesn't really sell our program very well. So, and they don't care what they've got as long as they're nice looking chestnut trees. So that's my idea, is to put some of these out in demo orchards initially, just to see how they do. And meanwhile, wait on Jared's analysis to see if we're okay at the B3. If we're okay at the B3 generation, then I don't have to worry about that, okay? But again, this is my insurance policy. In case we're not okay at the B3 generation, we've already got a step ahead on the next step, which is to put a little more Chinese in there, or to spike the punch. Yeah.
Okay, I have 2 things. I have a question, but I want to talk about what you just said about the male sterility on the F1. On the ones they're presuming are F1 contaminants, I would think they would have already looked to see if they're male sterile. It's pretty obvious. And
These are big trees, and they hadn't gotten up to look at it.
okay.
There's so many trees at Metaview, they can't look at it.
Okay, because I have male sterile, trees, and I know, that, you can see they don't fully develop the male.
Yeah, but if you're not, Mike, I'll give you an example for me. 'Cause, I mean, male steroids is what I did my PhD in, okay? I know what it looks like. But I got up, I have my plots, okay, in Carolinas.
Yeah.
I have 17 plots. Okay, one plot I have 2 trees that I selected. One of them is going down, looks lousy. I got up to pollinate the other one. I got up in the canopy of it in my 16-foot ladder. I'm looking around, and the darn thing is male sterile. And I said, what? And I start scratching my head. Why is this male sterile? And then I realized it was an accidental BC1. It was a seed that I had collected from a B3 tree, but there was an F1 not too far from it, a male fertile F1, that obviously was a pollen parent. And when I looked at it, I could see the Chinese features. All of a sudden, it made sense what this tree was.
Okay, another question goes back to the strategy of, recurrent, just doing recurrent, assuming that you haven't lost the same alleles in Nanking, in Clapper, and in Graves.
Right.
And the worry about inbreeding.
Yeah.
If you say you're starting with a certain population size of them, for example, here we can easily have 20 lines of 20 lines of Clapper, 20 lines of Grape, and 10 of Nanking. If you take 2 from each, it's 100 trees. If every generation you're doing that, you're increasing the number of trees that you're keeping.
Yes.
Shouldn't that be able to deal with the breeding issue?
It's a numbers game, and this is where Jared is a good quantitative geneticist. It's called calculating the inbreeding coefficient. There's an actual mathematical term, and he can calculate that and see if he's comfortable. But you can get from a common sense that that's what you have to worry about. Is you don't start out with such a small population that you wind up with an inbred population.
Right.
Okay. It's going to take a fair-sized population. Yeah.
So, did I understand that the pseudo F1 pollen parents existed in Meadowview, and some of that pollen was sent to us when we pollinated our wild American trees?
Okay.
Or am I at the wrong generation?
Where did the pollen of the pseudo F1s came from? That's what you're asking. Okay, remember these are open pollinated. These are from open pollinations. This is in Virginia, where there are huge Chinese chestnut trees in adjacent farms, and even if it's only 1% contamination, you're selecting for the most resistant trees.
Right. So my question is, at the chapter level, then, how did that affect us?
It means you better be very careful when you're selecting your good trees in your plots to look at them carefully and to send them off for analysis, if you want to, for the percentage, to see if some of them turn out to be 50% Chinese.
Right. Well, if they were, let's see, so the pollen, we got pollen from trees that may have been 50% Chinese.
Most of the pseudo F1s, my understanding, are B3F2s, not second or third back crops that you guys would have gotten pollen from earlier on. Is that correct, Paul?
That's right. Where these came in the B3
correct?
there were also contaminants in the B3 orchard, but they were usually easily picked out. There weren't as many trees there.
Well, we got B2F1 pollen essentially, and then we crossed back to America. So,
Contamination could happen in any generation. So
Right.
all I'm saying is just be careful. And if you're selecting for resistance, you're selecting for those outcomes.
yeah, but for us to have a pseudo F1, we would have had our pure Chinese times our local Americans. We don't have that. We don't have an actual F1.
Yeah, you may have, I've got a pseudo BC one, if you want to call it, that I found, okay?
Yeah, so we're not going to have as Chinese as Jared's chart.
Right.
So my question is, you might have more Chinese than 116, but maybe it's good enough still, even if it's a good tree. So we're talking about making things more Chinese. Maybe we're okay with.
That's the philosophical question I'm going to come to in a minute. Okay, so this, in my seed orchard where I presented this model, this is the 3 types of seed I'm producing. I'm producing B3F3 seed, which I collect from the B3F2 trees. Then I'm collecting what I call better B1s, which is an male sterile F1 times B3F2, and I have both Japanese and Chinese sources of resistance there. And I expect that these trees should have more blight resistance than the B3F trees. And I'm going to show you some data now, because I've got data on this. This is in 2018, this was a year ago, I made deliberate crosses between my B3F3 trees, and here's one right here, it's one of my selections, Clapper B3F2. And this is one of my male sterile F1s, which is right close to it. That was originally in a border, one of those borders of 10 trees. I've kept this one, which is a nice Nanking F1. It's male sterile. And I took pollen from this tree and I put it on that tree. In fact, I took pollen from 3 different B3F2 selections and put it on that same F1 tree, which I call a tester. And these are my data, and I hope you take a minute to look at this. This is thanks to Jared. This is from the small stem assay from this summer at Mediview. And what you have here is a genotype.
Paul,
Yeah.
before you go on, could you give a Reader's Digest on what small stem assay is?
Yeah, so, okay. The small stem assay is taking a group of trees, and he's been normally taking 40, and inoculating them with the EP155, the very virulent form of the fungus, when they're fairly small saplings, just a few months old. He's not trying to save these guys, he's just trying to see how fast EP155 grows in these saplings. And the idea is that you can, by looking at how a family performs, you can get an idea about how resistant the parent was. And those are, again, the data I'm going to show you, give you an idea. I'm trying to evaluate the parents by looking at the performance of their progeny. So he had Chinese controls in here from Pennsylvania. He phenotyped 33 of them, and 70% of those Chinese survived. This is pretty stringent test, and he didn't get 100% survival. American that he got, 29 of them, he had 28% surviving at the end of several weeks. I don't know exactly how many weeks. And then here's the data that I was really interested in. These are my 3 B3F2 selections from actually 2 different plots, times exactly the same Nanking F1 that I showed you in that picture. So I have one called prior 1-31, 1-79, and 9-148. That's plot 1, tree 31, plot 1, tree 79, plot 9, tree 148. So these are 3 of my Clapper selections. And what was really interesting to me, comparable numbers, was that one of them had much better survival, in fact twice as good survival as the other 2. So my initial assumption would be that this one is much better. In fact, these 2 don't look much better than American. There's also other information I have about these trees which leads me to believe these data. First of all, I also intercrossed 1-79 and 9-148, again by controlled pollinations, to make a B3F2, and there were only 19% survived out of that B3F3. And according to this, that's my 2 worst trees that I happened to cross-pollinate, and they gave me a lousy result. So that's another reason to think that those trees aren't so good. The third thing that came into play was 179, which I actually thought looked like a great tree. When I sent it in to Jared to have it analyzed for percentage American, 1-79 is 99% American. So that's another thing to make me think that I actually got some good results here that I can begin to depend on, that of these 3 selections in my seed orchard, that 1-31 is the one I should keep, and I maybe shouldn't even keep the other 2 except for some other purposes, which they've talked about maybe using them as parents times genetic engineering or something.
Paul, did you get, for 131, did that get selected and tested, the genetic test?
Yeah, actually I have not, off the top of my head, I don't think it's back yet. Because it was not actually as impressive a looking tree as 179 was. So I want to show you what these trees by themselves look like. Here's 9-148. That's what it looked like in 2018 when I did the pollinations. That's what it looked like this past summer. So it's collapsing. It's a tree right on the corner of the plot. And what Kendra and Sarah have told me is they find that border trees tend to do better just because of their position. So that we probably selected this just because of its position in the plot rather than the fact that it was such a great tree in terms of resistance. This is tree 179, and boy, this has been a kick in the gut for me because that was my prize tree. It's a gorgeous tree. And, you know, to know that it has not, number one, that it's 99% American, and number two, that it hasn't performed very well in my test cross, makes me want to cry. Because when I look at that, it looks like a great tree. And this is my warning to you guys when you start selecting. Watch out. This is one reason I like 179. You know, I had these little ideas come through my head. I thought one thing I might do is my grower doesn't like to have these lower limbs. So I trim the lower limbs, and of course that's a great place for blight to come in when you do this pruning. Well, this tree, 179, healed up all those pruning sites. Absolutely. No blight in any of those pruning sites. I was really happy. I said, this tree's really got resistance. You know, it can heal up all these pruning sites. But it's 99% American, and it doesn't do well in test crosses. So this tells me that this particular way of judging things maybe isn't so good. So this is the kind of thing I'm trying to think about when I'm going through, how do I pick out the best trees in my plot? Again, that's tree 179. Man, that was my prize. This is 131. This is the one that's supposed to be so good, and the test crosses are at least better. This is with the weekly strain of the blight. You know, it doesn't look that hot. It was better than some of the others, but the weekly strain, which is an intermediate strain between SG1 2-3 and EP155, is eating this thing up. So, again, that makes me think that our B3F2s still need some work. And whether it's intercrossing again with Nanking or Graves. I've only done those 3 selections. What I plan to do this coming summer is that I've got a whole bunch of selections now. I'm going to cross all those selections, if I can do it. I'm going to have to rent a bucket truck or something, on 2 different F1 trees to make sure that the F1 doesn't make a difference. These are from 2 different F1s from different backgrounds. Put them through the small stem assay and see if I come up with the kind of differences that you saw in that other data, a difference of a double, actually, in the amount survival. And if I can really distinguish between good trees and not so good trees by this test cross method. So again, that's one thing that I'm urging you as a Massachusetts chapter to think about, to put in these male sterile F1s, because they serve multiple purposes, and one of the purposes is to provide a tester for your B3F2s. Okay, yes?
You mentioned another factor was the fact that we're testing by punching holes and applying actively growing fungus to the infecting in that way, which is very different than the natural way trees would get infected by spores landing on the bark, and although possibly in the wound. So might we also have a problem because we're essentially not selecting for natural infection?
Yeah, Mike Novak has got a good point there. There may be more than one mechanism for resistance. And by actually drilling into the bark and putting in a plug, we may be overcoming one level. And that, I mean, I'm suspicious of that back with my, with my great tree there, that may have a type of resistance that does not show up when you punch inoculate it.
It may not even be a heavily Chinese one, because we essentially, since they're from American samples, our American population we've bred for might have a certain amount of bark resistance. We may already have selected by the fact that they were still around for us to sample.
That's right. There may be, but Mike, again, I try to think about how can I test for this. It's difficult. The punch inoculation is at least
Uniform.
Uniform, exactly. And so I think we can get an answer. Whether it's the complete answer, I don't know, but it's certainly an answer, I think, which is helpful, and I think it's proved useful. But it may not be the whole story. The question is, how would you test for that? I mean, it'd just be real difficult. Yeah.
A couple of points. First of all, I have surviving about 35 F1s that are all male sterile. I took Chinese pollen to an American tree. My tallest tree is probably about 30 feet tall. It's also the one that shows the best blight resistance. The other thing I've observed with these trees is that when the bark is still a smooth bark before cracking starts, if you look at the lenticels on these trees after about 2 or 3 years, you begin to see little orange dots right in the middle of the lenticels. And the tree's fine as long as that's it. That's obviously blight that's just sitting there with that tree and waiting for the splitting to begin. And when the splitting begins, it tends to line up between these dots that are going vertically. And that's where you get your blight problems going. Second thing, bark is traditionally a major factor in terms of protecting the tree. So when we do this corkboard test, as Mike just mentioned, for years now I've been saying I don't know that this is the right thing to be doing, because we're boring right down to the cambium layer and sticking it right in the cambium layer. Third observation I have is that the cambium layer makes tissue for the softwood. It also makes tissue for the bark. And it seems to me that the rate at which these two are going may determine how severe the splitting is and whether it gets down into the cambium layer. And I'm thinking that if the balance of growth for the softwood is way off compared to the balance of, or excuse me, I guess that's what I want to say. If the growth rate for this softwood is much higher than it is for the bark, then the bark's in real trouble, because it's getting, I have trees that are showing me a ½ an inch growth on each side after they got freed up, a tornado went through. And that's when the blight problem starts showing up. And it's clear that that softwood layer is growing at a tremendous rate compared to the bark layer. So I'm thinking that a third factor may be the imbalance of bark tissue that's growing versus softwood that's growing.
These are in essence pathology questions, and I'm a geneticist. I guess, in thinking about what you're saying, from my background and my prejudices, where I'm happy is when I can find a trait and I can find a consistent mapping location for it. If I can find a locus on a chromosome that consistently shows up with a certain phenotype, then I think it's real. And that's the way I think with Phytophthora. When I saw those 5 loci in all those different mapping populations in 2 different continents with 4 different species, then I begin to think that's real. I mean, to me, that's real. And so blight resistance may be complicated. It may have different factors. But if you can take at least one of those factors and get some genetics associated with it, then to me, from my background, that's real. It may not be the whole answer, but it's something. And we've got to breed for something, okay? So I know we've got a problem, but I think we're better than America. We've improved on the American chestnut. We've definitely done that. And the question is, how far can we go with what we've got? And we don't have the answer to that yet, but I think we've got the tools to do that. And I think in Massachusetts, you've got some great material to work with. So I'm really kind of excited about where it's going, and I wish I was 34 instead of 74, but that's
I've got you beat, I'm 80.
Okay, I understand. I'm supposed to have had an ancestor that lived to be 134, so I've got 60 years. That's real, folks, believe it or not. Okay, my conclusion. Okay, the controlled crosses to a common F1 tester is a good way of evaluating my B3F2s for blight resistance. It also results in better backcross seed for demonstration plantings, and as Kendra said, it spikes the punch. Okay. I'm going to turn a little bit to genetic engineering, which is, of course, now another tool in our basket. It could provide high levels of blight resistance, even higher than that of Chinese. Could provide a way, and again, for us in the South that are having to deal with 2 diseases, where we even have to add more Chinese genes, it's just really tempting to say if we could get a phytophthora gene, and they're actually working on trying to do this at Penn State, we could put in 2 silver bullets at the same time and solve 2 problems. And it could be a dominant, easily back-crossed to something easily explained to people, easy to do, and it's easy to get genetic diversity and local adaptation if you make enough crosses. If you only got 1 gene, it's a whole easier ball game, folks. You know that. So, you know, this is the way we do it. Instead of having that big diagram with all those pinks, you just cross a red and a white and you get a 1-to-1 a red to white. There's no pink anywhere. It's like the religion I grew up with. It was really black and white. Okay. Transgenic or cisgenic? This is one question that came up I addressed 5 years ago and I want to come back to. Some people are philosophically concerned about putting in a gene from a different species. So they say, well, if we could get the Chinese genes and put them in, that would solve the problem. We'd get it through regulation and we wouldn't have. Well, that's not true. A cisgene would be a gene actually from Chinese chestnut that you got and inserted. But this is my caveat down here. Cisgenic chestnut trees are not equivalent to backcross. So far, only 1 gene is changed in cisgenic, but there are over maybe 50 genes differentially expressed in Chinese versus American chestnut cankers. The cisgene is randomly inserted. That's actually changed in the 5 years. We now have some techniques called CRISPR and other things where we can actually replace a gene. So that's changed. We can actually replace a gene that we couldn't do 5 years ago. And the cisgene is under control of a DNA sequence called promoter that keeps the cisgene turned on at a high level in all areas of the plant. When they've done this genetic engineering, they tend to put it under a promoter, something that causes the gene to be expressed, which makes the gene expressed at a high level in all tissues. And that's not the way it actually works in the gene, in the tree. It's expressed in certain tissues at certain times. And to get that all straight in a transgenic plant is difficult. And then what do you choose as your transgene? What we may be breeding in is a switch, some kind of genetic switch that turns on a lot of genes. Whereas when you look at genetic engineering, they usually look at just 1 enzyme or something like that that's differentially expressed in Chinese and American in blight, and then they know what that is and they clone that in. But that may not be the entire answer that's turned on by the original switch that we would get in breeding. So it's difficult to pick out exactly what to clone and to put in from Chinese. Well, here's the potential problems. Well, TACF accused of bait and switch after 30 years of promoting backcross breeding. Well, we've just got to get used to it. It's a different tool. How will the public be able to tell the difference between conventionally backcrossed and GMO, if they care? The story of Johnny Appleseed went through the woods planting apple trees. I have a vision of Johnny Chestnut Killer, who comes in with a hypo hatchet, and which is one of these things you put in, Roundup or Garlon, and he goes, every chestnut tree is a GMO tree, and I'm going to hit it with my hypo hatchet to kill it, kill every tree in the woods because they're GMO trees. You just need one crazy to do that. So, is there kind of a labeling problem? Will only 1 or 2 make it through the regulatory process? They're trying to avoid going through the EPA for a couple of reasons. One of the reasons is the EPA will charge you $1 million per construct for them to evaluate it. They charge federal agency $100,000, they charge anybody else $1 million. And if we have to pay $1 million a pop for every construct we put through, that's a heck of a lot of money. They're evaluating now, will they allow fully fertile GMO chestnut trees to be released into the forest? This is a whole different situation from coming with a cornfield where it's only planted for one summer and you can dig it up or burn it up and plant something else the next year. If better types of GMO trees are later produced, how will they replace the earlier version? Are we going to differentiate them? And this is what I think Lois pointed out in her letter, are they being used as a precedent so that other more commercially important trees get through federal regulatory agencies? I think that's probably true, they are. But on the other hand, why not take advantage of it if it helps us? They're going to get it through one way or another, and why not ride on the back of the devil if it helps you get where you're going? Yes?
This is somewhat actually a legalistic question, I'm just very curious, ballpark answer if you can give it. How does the government, EPA or whatever, get involved in regulating? Is it through they're receiving federal funding, the people who are doing this?
Okay, he's asking how does the federal government get related? It's through specific laws relating, and with chestnut we have to go through 3 agencies because it's a food crop. We have to go through the food division. We have to go through the U.S. Department of Agriculture, that has one set of laws, and then the EPA has another set of laws. EPA is supposed to be against anything that is fungicidal. And what they're trying to argue with them is that this isn't fungicidal. The oxalate oxidase gene just prevents it from growing, it doesn't kill it. Whether the EPA will buy that is another question. But that's what they're trying to build. But it's specific laws. There's a FIFRA law, and I'm not the expert on it. Yeah. Yes?
So back in your previous slide, one slide back, in number 3, the last part of it, will this would like essentially one gene, the wheat gene, be too much genetic uniformity for blight resistance, which could then, with one mutation and one individual blight organism, which could then use its rather infamous ability to spread.
Yeah. This is an obvious problem if you only put, if it's uniform for one, it would be wiped out in one swap. And Yvonne, they're seriously thinking about this. We have some good people involved now. There's a lady that Lisa Thompson has recruited called Debbie Delmer, who is a member of the National Academy of Sciences, that's on our advisory committee now. She's very aware of this, very aware of how this happened in some other crops. They're thinking about putting the OXO, at least 2 different constructs of OSO, so that, because when you put that gene in, you're also dragging along some of its original American parent that it put in. It's also uniform for a certain number of genes on either side of that construct, just because of the recombination. And so you want to have it maybe from at least two different positions, so you're dragging along 2 different sets of genes. And then they're thinking again of maybe some Chinese genes. They're thinking of a lot of things. The question is how practical it is, and I don't know. And at this point, it's a lot of work, it's a lot of money, and I don't know, but they're thinking along those lines. And again, we don't know whether the GMO is going to be accepted or not.
Right. But, so, yeah, I just wanted to thank you for putting that in there and point out that.
Yeah, and I think, again, I can really support our scientific staff. They're very aware of these issues, very aware of these issues. Yeah. And this is the last thing I'm going to point in. You know, one thing, again, philosophy makes a difference. And one thing that's interested me about when we talk about how much Chinese you're willing to tolerate is that 3 of us who are willing to tolerate a little more Chinese all had training as plant breeders. It's Bob Leffel, the guy from Ohio. Greg Miller, yeah, Greg and I. We're all willing to tolerate more Chinese, and we were all trained plant breeders. Bob and Greg went to Iowa State, and I went to Cornell. That's our degrees are in plant breeding. And somehow we're a lot easier with Chinese. But the people trained in forestry, I'm maybe painting with a broad brush here, but some of the people in forestry, they tell me, if it's not 99% American, I don't want to deal with it, you know. They're really hype on this thing. It's got to be really American. And that's philosophy. It's partly about what you think will be adapted to the future. I think our environment is changing, and I'm not sure that, it's called the naturalist fallacy, is that this is the best of all possible worlds. That what's here is what's adapted to the present. And the present may be changing faster than the trees are. So to me, it's not guaranteed that having something 100% American is necessarily better than having something that's 75% American. I don't know. But it just is interesting to me that three of us who are less concerned about this all got trained as plant breeders, where we're used to crossing species and taking any tool out of the book. Okay, am I going over?
No.
Okay, so this again is the philosophical questions. What percent Chinese are you comfortable with? How comfortable are you releasing GMO into the wild? And do cis versus trans genes affect your attitude toward GMO trees? These are three philosophical questions that I see come up a lot in our discussions, and I don't think there's any right answer. I think you just have to decide for yourself where you land on the spectrum. And practical questions, I think these are three practical questions I think can be answered in the near future. That's why I put them up here. Number one, how many Chinese chestnut genes are involved in blight resistance? Jared is working really hard on this and has the money to do it. It's just a matter of time, and I think in a year or two he'll have some pretty good answers here for you. Number two, do we have enough resistance genes remaining in our B3F2 to warrant a recurrent selection? Again, I think we'll have the answer to that within a couple of years. Meanwhile, get it set up to do it, okay? Don't waste 18 years of work. And number three, will the OXO gene be approved to release into the wild? I think we'll know that in 2 years. So these are three questions that I think will be answered in the next 2 years, and I think they will all affect our program. Meanwhile, again, I think the Massachusetts, you're doing great stuff. I recommend putting in some of those male sterile F1s for the reason. Yeah.
On the second part of it, you're not talking about any individual trees, you're talking about within a population of them, correct? On number two there.
No, I'm talking in the population.
In the population. My background's not so much in the biology of it as it is in math. And that was one of the things that hit me as soon as this whole thing was announced. And so I did some kind of mathing. And the probability is rather low that we've lost any of them in the population as a whole. In other words, if you take, our clapper trees and our graves, all of them, 20 lines of, it's unlikely that the same ones have been lost in all of them.
That's true, but the caveat there, Mike, is that we started with one BC1. If we had started with the original Chinese, we'd probably have everything.
No, I realize that clapper may have lost.
Yeah, so I think within our population we have everything that clapper had. But the question is, did clapper have it?
But graves isn't populated clapper.
No. So again, that's the reason for combining graves and clapper and Van King.
Okay. When I say population, I mean all our population.
Okay, yeah.
If you're doing more analyses about our work, I'd like to point out that our growing zone in southern Rhode Island is now USDA number 7, which is the same growing zone as Kentucky has, which now has ink disease. So we are not necessarily immune, and I think that it would do us, that's my opinion, but I think it would do us very well to assume it's going to get here sometime.
Well, I'm glad to hear Massachusetts and Rhode Island have got graves seed orchards, and that you're finding out which ones have phytology. Yeah, again, that's your insurance policy. You better. Back 20 years ago, we had our first science review. We had an old Dutch guy, Hans van Buren, wonderful sense of humor. And Hans took me aside. He said, "Paul," in his real Dutch accent, "Paul, you've got to be very careful in tree breeding." He says, "You make a mistake, it takes you a long time to correct it." So I've always kept that in mind, okay? don't do one thing and then think you're going to do something else 15 years later. Let's do a few things in parallel. Have an insurance policy. Think about what we're doing. Review this every year. You know, and I think you're doing it. I'm really happy again with the way you're thinking. I know, Jamie, you've got some good brains in this outfit, and use your brains. That's all I'm asking. Okay. Yeah.
Does soil pH come into play as a factor in the ink disease?
Not pH, except for extremes. If you get to 4 or 3, where it's acid as heck, it does not. The only thing that seems to come into play is temperature. That phytophthora in frozen ground doesn't do very well. So that's why you haven't had it in the north as much. You can thank that snow out there. Because if the climate is warming, the phytophthora will probably be moving north.
Oh,
Yeah.
so on a couple of slides back, you talked about your problems. And all the problems you talked about with GMO were more political problems. And then you talked about, you know, the slide about, the slide before talking about the actual practicality of this particular GMO tree and whether it's there's opinions about whether it's a good idea to put it out in the wild. So I'm asking your opinion, what's the effect of actually, do you think it's a good idea, bad idea, or if you're not sure, what's the worst, I don't know.
What's the worst thing we could do with an OXO tree, right? I mean,
What's the worst effect? There's a lot of concern about GMOs, and we're trying to get a sense.
they've looked at pollen, they've looked at tadpoles, they've tried everything. They can't see it. I don't know. I remember there used to be a real good program on genetics by David Suzuki, who was a professor at British Columbia. And they asked David about genetic engineering. He said, there's going to be some bad things, but he said it'll probably come out of left field. He said it'll be something that we're not expecting. And to me, it's like atomic energy. There's some real benefits to atomic energy, radiation. People have been saved from cancer. And there's also some real problems. And I think that's with GMO. It's potentially can cause. I mean, the thing that's happened already, and 30 years ago when I was at Cornell, we knew it was going to happen, was Bt corn. One of the things for corn, they're putting in herbicide resistance and insect resistance. An insect comes from Bacillus thuringiensis, this bacterium that produces a crystal that kills insects. And there's different forms of Bt. It kills different types of insects. And what they did was to put in one Bt gene in corn against European corn borer, which is a big problem in the Midwest. And it held up for a while, and then it went down. The corn borer developed had a gene that overcame it. And I minored in entomology, and our entomology professor said what they should have done was to put in 2 genes at one time, 2 different genes, because then you would have had to have a European corn borer that had 2 mutations at the same time to overcome that resistance. If you put them in one at a time, all he has to do is have 1, he overcomes that, then you put in number 2, then he gets in 2. If you do them seriatim, it's easier for the pathogen or the bug to overcome it. If you put them at the same time, he has to have both genes for resistance at the same time, and the mathematics of that are much more difficult. But from a practical point of view, the companies didn't want to wait to put in 2 genes. Okay? So we knew it was going to happen. It happened. Yeah.
Yeah, I just wanted to kind of back up what you just said, but I had my hand up already. But yeah, Jamie, it's not just politics. There's scientific reasons not to just jump into the OXO basket by itself. Those arguments don't necessarily hold up the same way if you talk about combining the OXO gene with our partly resistant Chinese-derived trees. So that's combining the 2 sources of resistance, although one might not be well developed right now, and then its signal would be making target spot. So it's complicated.
Yeah, but it certainly is. I mean, from a breeding point, perspective, it would be wonderful to have a single dominant gene. That's what my buddy has in Hazelnut. He's got a single dominant gene for Eastern Philly. But he has to breed for 24 other traits to make hazelnuts commercially viable. This is the only trait we have to worry about.
But I worry about people seeing an NXO-derived tree look really pretty for like 10, 20 years, and then we get like forests of them.
But Yvonne, you have to admit, you're not really comfortable with GMO, right?
No, I'm not. It's not like, I'm not like totally against it if it were actually studied and considered in an ecological context, like what you're talking. It's more, and the thing is that in Europe, they have a better developed scientific process to evaluate it, whereas here, a lot of that seems controlled by people with financial interests in it. So, you know, I like the European approach to it. You know, they're not all, it's not like, you know, anti-vaxxers or something, you know.
Yeah. Each person, I think, has to answer that for themselves.
Yeah.
And meanwhile, I love these McCown apples at Cornell. We used to go to the apple store every fall. That was my favorite apple. And when I saw that back there, I said, yay! So all of you pick up some apples. They're really good.
They're available for people to take home. I don't want to take any back with me, so please help yourself on the way out.
And Yvonne, the other thing I always think about oxalic acid is, you know what plant has lots of oxalic acid? Spinach. And as a kid, I was forced to eat spinach because my mother thought I was going to be Popeye. She forced it down my throat with a fork. So, you know, I have sort of a bad feeling about oxalic acid. It binds up calcium, it gives you osteoporosis, it's nasty stuff. So if there's something that overcomes oxalic acid... Yes.
Dr. Paul Sisco, I really like where you're going with, you know, the more chestnut. Because, you know, as planting trees in the forest, we're just interested in a good tree, not the perfect tree, but a good tree that can compete and grow in the forest. And I just, I like the direction you're headed. And I think that will give us what we need.
Again, I think you've got a great thing going in Massachusetts. You've got a wonderful coordinator with Kendra.
Just $20 later.
Pardon?
She'll give you the $20 later.
Okay. Well, she's got a system.
220.
No, she really is. She's good. And just keep up the good work, folks, and put in a few male steroids once now.
Thanks so much.
Thank you very much. So this is the time to schmooze. So please feel free to sit and talk, and thank you all so much for coming. And you can now corner Paul and ask him all your questions.